Hi, I'm Saraswati Gajapati Rao from SMK Yukwan Batu Gajah. Today, I'm going to discuss on a topic from Biology Form 5 KSSN under the theme Inheritance and Genetic Technology, Learning Area Genetic Technology, Content Standard 13.2 Biotechnology, Learning Standard 13.2.2 describe applications of biotechnology in life. The learning objective: at the end of the lesson, you must be able to describe gene therapy and describe DNA profiling. I'm going to focus on two applications of biotechnology in life, which are gene therapy and DNA profiling. Now, let us look into gene therapy. Gene therapy is used to treat or prevent genetic diseases. In gene therapy, a normal gene is inserted into the patient to replace the abnormal gene. The diseases that can be cured by gene therapy are cystic fibrosis and a few types of cancer. These are the basic steps involved in gene therapy. First, a cell with an abnormal gene is identified. Then, a normal gene is inserted to replace the abnormal gene using a virus as a vector. Then, the cell will finally perform normal functions. CRISPR. It is pronounced as CRISPR. Clustered regularly interspaced short palindromic repeats. What is CRISPR? CRISPR is a technology that can be used to edit genes. CRISPR or CRISPR-Cas9 is a unique technology that enables geneticists and medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. How does it work? The CRISPR-Cas9 consists of two key molecules that introduce a mutation into the DNA. An enzyme called Cas9, which acts as a pair of molecular scissors, is the first key molecule that can cut the two strands of DNA at a specific location in the genome. The second key molecule is a piece of RNA called guide RNA. It consists of a small piece of pre-designed RNA sequence located within a longer RNA scaffold. The scaffold part binds to DNA and the pre-designed sequence guides Cas9 to the right part of the genome. In the diagram here, we can see how the guide RNA will guide the Cas9 to the right part of the genome. Let's look at how CRISPR-Cas9 is done. First, the DNA with a target sequence is identified. The guide RNA will bind to the target sequence. Then, the Cas9 follows the guide RNA to the same location in the DNA sequence. The Cas9 enzyme then cuts both strands of DNA. At this stage, the cell will recognize that the DNA is damaged and it tries to repair the damage. The cut is repaired and this will introduce mutation. Scientists can use the DNA repair machinery to introduce changes to one or more genes in the genome of a cell of interest. Now let's look at the second application in biotechnology, which is DNA profiling. DNA profiling is a technique used in forensic to identify individuals based on their DNA. This is due to the fact that an individual's DNA is unique, except between identical twins. An individual can be identified based on the DNA obtained from blood, semen for male, or skin. The applications of DNA profiling. It is to identify criminal, to identify genetic disease, to settle paternity and maternity dispute, to determine suitability of organ donor and recipient. These are the steps used in DNA profiling. First, the blood, semen or skin sample is obtained from an investigation center, example, a crime scene. Next, DNA is extracted from the sample. Then, restriction enzyme cut the DNA into different sizes of DNA fragments. The DNA fragments which have been cut will have different sizes and then they are separated by gel electrophoresis. The DNA fragments of different sizes are then transferred from the gel to a nylon membrane. Radioactive probes are added to the nylon membrane. X-ray film is then placed on top of the nylon membrane. X-ray film is then processed to show the position of DNA bands that form the DNA profile. Let's look at this section. Think smart. Try to answer the question below. Based on the DNA profile below, can you determine who is the biological father of the child? So we can see that there are 4 DNA profiles here. Mother, child, father 1 and father 2. We are required to find out who is the father. Father 1 or father 2. Let's look at the DNA profile. The DNA profile of the mother and child matches at 1, 2, 3, 4, 5. 5 fragments. If we compare the DNA profile of child to father 1, there is 1, 2, 3, 4, 5 DNA fragments that matches. Okay, let's compare father 2 with the child. Okay, for father 2 there is 1, 2, only 2 DNA fragment that matches. So we can conclude that Father 1 is the biological father of the child. Now, let's look at another question. Let's read the situation. After a father who was a millionaire died, there was a property dispute between four siblings. A knife with traces of blood was found by police at the scene. By using the DNA extracted from samples of knife blade, victim and suspects, the following is the DNA profile obtained. So we can see that there are victim, the sample of blood from the knife blade, suspect 1, suspect 2 and suspect 3. So there are 5 DNA profiles. Okay, as one of the forensic expert, how do you handle the case? How do you handle the case and convince the family involved about the murder suspect? So, for this question, you have to explain how you will do the DNA profiling. That means, what are the steps of DNA profiling and who is the suspect? How do you convince the family? Let's identify the suspect first. So this is the sample obtained from the crime scene. Okay, let's look at the victim and crime scene's DNA profile. Okay, so we can see that the first fragment matches. Okay, there's only one fragment which match. Okay, let's look at suspect 1. Okay, there's 1, 2, 3 fragment that matches. Let's look at suspect 2. There's 1, 2, 3, 4, 5 and 6 fragments. Let's look at suspect 3. There's 1, only 1. So we can conclude that the murderer is suspect 2. So the sample taken from the crime scene matches with suspect 2's DNA profile. So we have found the murderer. Let's look at the answers. Okay, so we can tell the family that the criminal is suspect 2. So you get 1 mark for that. Then explain how the DNA profiling is done. Restriction enzyme is used to cut the DNA into fragments of different sizes. The fragments of different sizes of the DNA is separated by gel electrophoresis. The DNA fragments are then transferred from the gel to the nylon membrane. An X-ray film is placed over a nylon membrane. The X-ray film then processed and DNA profiling are generated. The strip arrangement of the suspect's DNA fragment corresponds to the DNA found at crime scene. That's all. Thank you.